Ladies and gentlemen, please welcome Program Director of In-Space Biomanufacturing at the ISS National Lab, Davide Marotta, to the stage.
Walking on a dream. How can I explain?
All right, thank you. Good afternoon, everyone, and I hope everyone is having a great start of here at Ascent. And hopefully you got a chance this morning to hear all the session and hear of our astronauts on station talking about the work they are doing, which brings to this afternoon session. And obviously, biomedical and regenerative medicine research in LEO is like helping better to understand how we can live effectively off planet, but also to bring terrestrial benefit to us. Now, obviously, I'm a little bit biased on that because I work for the ISS National Lab and I'm the program director for in-space biomanufacturing. So, you know, my everyday job takes me to interact and evaluate proposals and work with the research teams that they're doing actually research on station. So while I'm an ardent believer in the notion that space can unlock secrets that can prove useful to us for future R&D, this session also brings together a variety of highly influential researchers and partners to showcase a little slice of what the space station in low Earth orbit can bring us and can do to the biomedical community. We will have the ability today to hear from how government is supporting biomedical research in LEO, how hardware partners are a critical part of ensuring science viability. And then we also hear from researchers themselves who have either conducted a variety of experiments on station or are embarking on the space station. And lastly, we would like to hear questions from you, the audience. But so with that, while I could, you know, read everyone impressive bios, like I figured it might make sense to head down the line and have themselves to introduce themselves. So let's start with the first.
Okay. Hello, and thank you so much for inviting me to be here with you, Davide. And it's a pleasure to be here at Ascend. It's my first time with this audience. I am the director for the Biological and Physical Sciences Division under the NASA Science Mission Directorate. I hope many of you were able to hear Nikki Fox this morning. And I will pass it off because I'll talk about my division later.
Hi, everyone, and thanks again for having me as well. My name is Stefanie Countryman. I am the Director of BioServe Space Technologies, and we are one of the implementation partners that helps researchers, companies, other organizations translate their ground-based research to a successful spaceflight research program.
Hi, everybody. I'm Arun Sharma. I'm a professor at Cedars-Sinai Medical Center in Los Angeles, California. I've had the opportunity to conduct multiple independent investigations aboard the International Space Station, examining the impact of microgravity on stem cell function, as well as the effect of microgravity on the individual cells of the human heart. And it's great to be here. It's my first time here at Ascend.
I'm Jing Lee. I'm a professor at the Wake Forest Institute for Regenerative Medicine. So yes, I'm also first time here. Very excited to be a part of these panels. And we do bioengineered tissue and organ construct for treat patients. So I'm sure that this technology could be beneficial not just only in the Earth and also people, human in space as well.
Awesome, thank you so much. But let's start with the first question for you, Lisa. Biological and physical sciences, but obviously today we are more on the biomedical side. Can you talk about how NASA values biomedical research and how the space station has played a critical part in assisting this mission?
Yeah, sure, and that's actually a really easy one for me. So in the Biological and Physical Sciences Division, our mission is to pioneer scientific discovery, enable exploration, and then return all of that technology development to benefit humanity on Earth. And I have to say that we do ground-based research, a very little of it. Our predominant laboratory is the International Space Station. We lead the world in fundamental space-based research. So for all of that very pioneering effort to studying the influence of microgravity and that spaceflight environment on biological organisms, from plants to yeast and to stem cells that Arun will talk about later, has all started with our division. And as far as the benefits that we're seeing through the space station, we started way back in the shuttle days and even before that, looking at different organisms. Today, we actually have a fully functional operating laboratory that allows us to look at things in ways that weren't possible before. We can do this in a very end-to-end, which we're looking at even advancing it further so we can complete an experiment in the space station environment. So has been a true value and a return on investment to the taxpayers.
Also, and big news came out last month about the national academies and about the decadal survey. For those unaware, can you provide some context on what this survey is and how it will impact like future NASA partners in low Earth orbit?
Yeah, so the Science Mission Directorate relies heavily on what we call the decadal surveys that are commissioned by the National Academies of Science, Engineering, and Medicine. And through that, they help to lay out the next decade of science plans for each of the Science Mission Directorates. And we were fortunate enough to receive ours on September 12th of this year, and we have a lot of work to do to figure out what kind of strategy that we're going to develop to accomplish the recommendations and the plans set forth in the decadal survey. And one interesting aspect of how it will impact our researchers and the commercial community is that it spans the time not only through the life of the International Space Station, but into that transition period to the commercial LEO destinations. And so as we're thinking of our strategy, how we'll get that research done and continue in low Earth orbit, we take into account in our strategic plan the folding in of those commercial LEO destinations and all of the commercial partners, whether it's hardware, whether it's the platforms, and then of course we're also always looking out to beyond LEO and the next exploration path forward.
Awesome, thank you. I would like to jump to, I have a question for you. Arun, you're an experienced flyer and launching your first beating ourselves project while you were a graduate student at Stanford in 2016, right? You have since flown multiple investigations on the ISS, and now you're post at Cedars-Sinai. Can you talk about your research and maybe even take a step back and how space came into the equation for you and your team? And I can relate to your experience.
Sure, happy to talk about our work. So you're right, I got started in this field as a graduate student at Stanford, but even before that, I grew up in a small town called Huntsville that some of you may be familiar with, which has a very strong space influence, obviously. When I was a graduate student, we had the opportunity to write a proposal for stem cell research in microgravity and to see how we could harness some of our model systems, perhaps to understand the effect of low gravity on the cells of the human body. And my laboratory at the time and also the work I'm doing right now focuses on stem cell-derived heart muscle cells. So these are called cardiomyocytes, the individual cells, heart muscle cells that are found in your heart, and they're responsible for the contraction of the heart. And what we're able to do is mass produce human heart muscle cells by the billions in our laboratory at Stanford, and actually now also in my laboratory at Cedars-Sinai. And we thought this was a really useful model system to understand the impact of microgravity and spaceflight on the individual cells of the human heart. So this is our model system to understand that. And so with a highly collaborative effort, and in large part thanks to Stefanie and your team as well, we're able to launch an experiment to the International Space Station in 2016, examining the effect of microgravity on these stem cell-derived heart muscle cells. We examined how the cells contracted, how they changed in their ability to beat in orbit, as well as conducted a number of gene expression, genetic analyses on the cells as well. Our eyes and hands in the sky were Dr. Kate Rubins, a NASA astronaut who is a very instrumental in helping us accomplish our experimental goals. And fast forwarding a few years down the road into, now I have my own laboratory at Cedars-Sinai, we're actually interested in taking some of these studies to the next level and thinking about ways that we may be able to harness microgravity for stem cell biomanufacturing. So we actually have a grant from NASA that's exploring that right now. We had a project that was launched on the AX2 mission just a few months ago where we were conducting the transfection or DNA introduction of a DNA into stem cell populations aboard the International Space Station, eventually for down the road, potentially manufacturing of stem cells and products on ISS.
Awesome. At ISM, Jing, like you and your team at Wake Forest Institute for Regenerative Medicine you're about to embark on your initial vascular tissue challenge payloads to the space station next year. So can you talk a little bit more of this vascular tissue challenge?
Sure. I think that I can share my slide, and then maybe better explain what we do and then how we involve in vascular tissue challenges. Can I have my slide?
Should be.
So we actually do, as I mentioned, that we do bioengineer tissue and organs that is similar to native tissue organs. So they could repair damaged tissue and organ that construct. So we do these researches many years, and then suddenly NASA created a vascular tissue challenging. That was back in 2016. And vascular challenging BTC actually request. So this is, this slide is showing that we do engineer many different tissue and organs, that including bone and cartilage, and also complex, also chondral and muscle tendon. And also we also target to engineer solid organ like heart and liver and kidneys. So this slide actually shows that we utilizing 3D printing technology to manufacture tissue and organ-like construct that containing live cells. So we printed live cells and making 3D tissue architecture that is mimic that native tissue structure and anatomies. So, but engineering tissue, one of the challenging is vascularizations. After implantation, this construct should be vascularized immediately. If not, even though you fabricate constructs very nicely in vitro outside of the body, but after implantation, they may be not survival making and function eventually. So vascularization, one of the challenging in this field. And can you go to next slide? So vascular tissue challenging that create from the NASA that requests creating thick vascular human tissue organ that showing metabolic functionality over time that requires 30 days in vitro survivals. So in order to achieve these goals, we are utilizing 3D printing technology, making a perfusible liver construct that containing liver cells. And then we are able to show integrate with perfusion system with our engineered liver construct and showing their viability up to 3 days in vitro, as is shown here. And it is not just only survival, we also look at the liver function like albumin production and bilirubin production that is very similar level of human physiologies. So we luckily successfully complete this trial in 2021. So as I mentioned that it started 2016, but we complete 2021. It's a 5 years long journey, but this could provide an opportunity to integrate with NASA. So now we are more involved in space experiment using our technologies.
So originally it was a ground-based project. How launching your experiment to space shifted the way you and your team has to think about the logistics and about the experimental plan?
Right, so engineering a tissue organ definitely beneficial for the patient in Earth, but also that could be a good model system investigate effect of radiation and gravity in space as well. So, and also, we are also very interested in biomanufacturing as well. So biomanufacturing and tissue and construct that can manufacture in the space and then back to Earth for patient treatment. So I'm sure that we will talk about in detail, but in space manufacturing, especially utilizing 3D printing could be very beneficial.
Awesome, thank you. And Stefanie, BioServe has a long and rich history of supporting both NASA, international partners, and ISS National Lab life science investigation on the station. Can you talk about how your team originally got started first and outline the importance of having a trusted and effective hardware partner if you are a prospective space researcher?
Sure. Well, BioServe actually started via NASA grant back in 1987, so we've been around quite some time. We were established as a center for the commercial development of space. So even way back then, there was some thinking around commercializing low Earth orbit. And we specialize in the life sciences. So at that point, when we were really established via two universities, the University of Colorado in Boulder and Kansas State University. And the university in Colorado was the engineering group, and the university in Kansas was the biology group. And so at that point was really when we became a multidisciplinary center of having both science and engineering expertise to support non-traditional users of space who could not only, the research that they do could not only help exploration, but also help benefit people here on Earth. So it was really kind of ahead of its time, I think. And it's nice to see that come full circle with the ISS National Lab and the commercial low Earth orbit destinations. But it's really important to have a group that understands how to translate ground-based experiments into a successful space-based experiment. There are a lot of things that happen, obviously, in a microgravity environment that people who work in the ground don't really think about. Like when we talk to researchers, you know, you ask them to explain how they do their experiment on the ground, and they can tell you all of the details, and probably 85% of those details are gravity dependent. And so understanding how to translate that into a successful spaceflight experiment, and also follow all of the guidelines and regulations that we have to in terms of safety and safety of the vehicle and safety of the crew when we're conducting things on space station are really our areas of expertise, and working closely with those science teams to help develop an experiment that can be successfully conducted but also closely resemble how they do their research here on Earth is something that is, I think, really important, and it really helps for quality, it helps for teams to have quality and impactful research results.
Good, thank you. I can totally relate, because when I started, you know, I was at the bench doing my science research, and when I started, you know, working with those guys, I was completely lost. Like, but, you know, the guidance and now we provide to the new researchers that we use microgravity is fundamental. And also, there is another aspect that this is more a question for Lisa. When I meet a potential researcher that when I use microgravity and have access to the International Space Station, they're kind of confused on the role between NASA and ISS National Lab in supporting the biomedical research. So how would you characterize the similarities and differences between the two?
Yeah, and I get that question often myself, and you know, I think in some regard we see us working together as one, but we are separate entities where, you know, NASA, we're both using the space station to advance science in space and understand that the NASA mission is to look at science up in space. You know, we have the fundamental aspect, but also enabling exploration. So how can we learn from that to help protect the crew for long duration missions or for sustainable presence on the lunar surface? So a lot of the research that NASA does is for the NASA missions. Now, with that said, a lot of the technology and science that we learn in our missions gets translated to benefit life on Earth. Whereas for the space station, the mission of the ISS National Lab is to bring in the commercial partners, to bring in that community, the other government agencies, for the distinct benefit of life on Earth, and they do a great job of that.
Thank you. And there is always occasion to collaborate, and we are collaborating on a recent research announcement, Igniting Innovation Science in Space to Cure Disease on Earth. Do you want to talk about this collaboration and this new research announcement?
Yeah, so believe it or not, this is one of the firsts for NASA and the ISS National Lab. Who would have thought that, together we do have common intersections and, leveraging off of the International Space Station National Lab's mission for all of that research that's being done on the space station to benefit life on Earth. And that is something that is very important to NASA. But then also combining that with not only what we're doing in that space station environment to benefit life on Earth is how does that benefit NASA as well? So we found this beautiful intersection called Igniting Innovation, and it was space science to cure disease and health for Earth. And those are things that are critical for our astronauts and crew going on these deep space missions. So it was a perfect intersection and very exciting opportunity. We just had step 1 proposals come in, and we're really excited to see what comes in for the step 2 and awards.
It's absolutely thrilling. We are so excited at ISS National Lab as well. And looking at the biomedical research that we do on station, researchers need implementation partners. We have Stefanie BioServe here, but also a question for you, Stefanie. Many more have appeared on the stage, implementation partners, to support life science on station. So how has the addition of these companies and their facilities benefited the space station program, or let's say the space station program, please?
So the space station just wouldn't even exist without, and the research that we're doing without the implementation partners like BioServe and all of the other commercial partners. There's only so much bandwidth that NASA has, and developing this hardware, as Stefanie was describing, to do an experiment in space is really exciting and really challenging. I'm sure Arun can, and you can speak to that. But to be able to reach out to these experts now, we didn't have this many years back. These experts have that experience. They can help guide you through the process, make it less intimidating, and they know a lot of the things you don't even know that you need thus far. So they really help to guide the researchers and make their experiments a success in that space environment.
Yeah. I call the implementation partners the fairy godmother of space, because they guide you.
That's right. They do.
Don't worry, I'll take you there. So, Stefanie, you have been, BioServe has been around for a while, and in your eyes, can you describe how you see the evolution of the space station from its earlier days?
Mm-hmm. Yeah, it's in the early days of station, the focus, and rightfully so, was on assembly and getting it built and operational. And so in the early days, and it was even before the ISS National Lab was developed, you could do some science, but there was limited amount of science that could get done. There wasn't a lot of crew time because they were busy doing assembly and putting the systems together. And so since assembly has been complete, and since the advent of the ISS National Lab, and also with the growth, in my opinion, of the BPS research, the amount of science that can get accomplished on the space station now is significant, and what has been done is significant, and the capabilities that we have, not only through our organization, but all of the other implementation partners, as we spoke about, and a lot of other commercial entities who have put platforms on the space station. The amount of research that's done is amazing to me, and the variety of research that's getting done. And and just the variety in life sciences. I mean, back in 2001, I don't know, at that point I would have envisioned supporting experiments that are, tissue engineering or functional heart tissue or organoids or organ on a chip platform. So the variety of the types of science that can be done is just amazing, and it keeps growing every day. And in my opinion, that's only benefiting the NASA mission, but it's also benefiting, people here on Earth as well.
It's a constant evolution, and you, Arun, you have worked and sent payload in the traditional way, let's say, but also you have collaborated with commercial partners like Axiom Space to Launch projects on the private astronaut mission. Are the process much different, or how would you describe?
Yeah, honestly, it was really comparable in terms of the experimental preparation upstream, in terms of the actual experimental manipulations that the astronauts were able to conduct on the station. I don't think there were, from the principal investigator point of view, I personally think they were pretty interchangeable, which I think is a good thing. I don't think necessarily you want to have a difference in experience between the two options. I think one advantage that the commercial side might be able to provide is down the road having more mission specialists or technical expertise astronauts. Say if I want to do a very specific type of stem cell experiment on the International Space Station where I had to manipulate certain heart tissue or so on, perhaps a commercial entity would be able to fly a mission specialist who is an expert in stem cell derived heart cells, right? Maybe they could fly me up there, I don't know. But no, I think for the large part, these are interchangeable experiences, which is a good thing.
And speaking of processes, and we have also Stefanie can speak about processes on these. You are a researcher, obviously, and what's the process like to submit and then develop the entire project, like from the early stage, let's say?
Yeah, these are some of the most this is the most collaborative area of study that I've worked on in my career, because we need a team of not just biomedical researchers, but also engineers, aerospace engineers, mechanical engineers, folks who have a lot of experience in doing this type of work, such as the implementation partners. We rely on the implementation partners very heavily for our pre-flight preparations, just to ensure that the work that we want to do can be done aboard the International Space Station. Certainly there are certain technical restrictions, certain safety regulations that we have to meet with all the experiments that we conduct. To simplify, and an example of this is actually shown on the screen. In our laboratories on the ground, we can grow our cells in what's called 6-well culture plates, but we can't do that aboard the International Space Station because if we take the lid off of one of these plates, the cell nutrients are going to escape into the space station, and of course no one's going to be very happy when that happens. So we have to collaborate with the implementation partners to design custom pieces of hardware, such as the one in the bottom right corner there that astronaut Kate Rubins is holding, that would actually facilitate the growth of the cells that we're interested in researching aboard the International Space Station. But, and this is something that was reflected earlier, the space station is an advanced research laboratory, and I was actually, the first time I did research in space, I was very impressed by the capabilities that were found aboard the ISS, because you can see advanced microscopy setups, you can see incubator setups that we would have in our laboratory, you can see a lot of the same reagents and pieces of equipment that we would use on the ground. But in general, this is a very highly collaborative effort, and we need everyone, such as the implementation partners, all of our partners, to get these sort of experiments done.
Cool, thank you. So it's a lengthy process, but I guess the juice is worth the squeeze, like it's worth it. So and we are experiencing that every single day. So Sanjay, for you, I remember a few years back when the Redwire team launched and validated the first 3D bioprinter to the station, talking about biofabrication. A lot of people felt that it was a major development for those interested in living and working in space, for example. But now the notion of bioprinting human tissues and organs seems literally like science fiction. It became like science fact. When you think about bioprinting and tissue regeneration in LEO, in space, on board of the station, do you pinch yourself?
Sure, yeah. So, yeah, bioprinting, 3D printing is very popular these days. Everyone knows 3D printing, right? But now when we start 3D printing, it was really new. But we thought that this technology is very clinically relevant because laboratory setting, you cannot creating very complex structures. But 3D printing, you can manufacture tissue construct with complexity. But at the same time, we have very limitation on the ground, so in terms of the process. So you can simply think about the 3D printing process. So we printed the cells. So in order to print the cells, we are using hydrogel systems. But we use that very viscous, highly viscous material in order to maintain cell suspension homogeneously before printings, right? And then this cell-laden hydrogel is passing through very small, fine nodules, and then build that 3D architectures. So we need a rapid cross-linking process to maintain their 3D tissue architecture. If not, this structure could collapse immediately. So on the ground, there is lots of limitation. But I'm thinking that in space, in terms of bioprinting process, there is no gravity. So we don't need high viscous materials. So that means that we can use low concentration of hydrogel system that containing cells. And then when you printed structures, maybe we don't need a rapid cross-linking process, maybe, right? So there are, even though I have not done 3D printing in space yet, but I see there are many advantages utilizing 3D printing technology in space that may be very promising to me as well.
Cool, thank you. And soon you're going to have the possibility to go to the Kennedy Space Center and see the fire and the launching, and you will get your experiment back and ready for look at the results and how it went up in Leo. So that's pretty exciting. I'm sure in the audience we have some prospect researchers that are thinking, oh, maybe I should take my R&D to Leo. So Stefanie, what are the main recommendation that you would communicate with the researcher team that are considering to use microgravity for that R&D? What would you say to them?
Test early and often is what
Absolutely.
I would say that you need to work closely with whatever group is helping you translate your experiment into a space-based experiment. And I would say that there's a lot more testing involved to ensure a successful experiment than you might actually realize or think. And so a lot of the groups that we work with, kind of partway down the path of getting ready for a space flight, they typically comment like, I didn't realize how much testing would be involved. But that's needed in order to have a successful flight experiment, because you are doing things. We do try to use some COTS, commercial off-the-shelf hardware, but as Arun mentioned, there's reasons why we can't use all commercial off-the-shelf hardware. So you're going to use hardware that's a little bit different than you're used to using in your lab, and so that requires a lot of testing to ensure that that your science is going to react the way that you would expect it to once it's put into hardware that's certified for space flight. So I think my biggest recommendation has always been, you need to really do a lot of ground-based testing to prepare you for your flight. And really that testing should culminate in a full-up dress rehearsal that everything is done exactly the way that it's going to be done on station. It's just not on station yet. Yeah. So I think that's my biggest advice to any potential researchers.
No, absolutely. And like the overall process, like how long does it take more or less? Like from concept development, hardware configuration, and testing and launch and return of the experiment and data analysis for the researchers. Like, can you give
how long does it take?
Yeah, an estimation. Yeah, because I'm sure people are wondering like.
Yeah, it does depend somewhat on the complexity of the experiment and the type of hardware and the type of science. But on average, I would say right now we're at about a 12-month from the time of award to the time that you're ready to fly. There are instances where you can go faster, But even the process itself it takes a while to get through the process, like all of the documentation that needs to be done and the paperwork that needs to be done in order to be approved to fly something to the space station. So, and that's something I think will be interesting to see as the commercial platforms come online, is if that process could be streamlined a little bit and maybe make it a little bit faster. But there's still a lot of testing that I don't think you want to rush through. So most of the time, and particularly with life science experiments that we're involved with primarily, it's just that the complexity of them require time to test. So I would say around 12 months is probably the average length of time from award to the time you're ready to fly.
Cool, thank you. And we had commercial LEO destination. My question for Lisa now is like, the station is planned to continue through 2030, more or less, and possible beyond the date. But what are some of the main areas that you and your team will support in terms of life sciences and biomedical community to ensure like the transition in the future into commercial LEO destination?
Yeah, so that is a question that's been, on our science community's mind since, we learned about the lifespan of the space station, and that we actually have a target and we have awards out, phase A or phase 1 awards for the commercial LEO destinations. And there's, been a lot of concern. What is it going to look like, as Arun was talking about, which I think is a great example of, working with the commercial provider was pretty seamless. It was very similar to working with NASA. And so how is that going to look in this new future commercial LEO economy and platform? And I think NASA is working that. They're working closely with the commercial sector. We work with them on capabilities that are going to be needed to conduct all of the life science research that's really important, not just to NASA, but to the entire science-based community. And we're also working on different strategies for helping how we move the scientists through that, because right now they get to come to NASA or ISS National Lab, and we basically help facilitate that process. And we're working with the commercial LEO destination platforms to see how we can still maintain some sort of infrastructure similar to that, so that it remains seamless, as Arun was mentioning earlier. So we are at the forefront of those discussions, so you can be assured that we are working to have a seamless process.
And when the time comes for the station to be retired, what do you think is the legacy will be specifically for the life sciences?
Wow, that's a really huge question. And to identify a single legacy, I think would be too much. But what I can say is that I do believe that one of the biggest legacies that we will have demonstrated on the space station once it ends is the ability to live and work in space for extended duration. I'm not sure how many of you have followed along, but from the shuttle era all through space station, I can remember the first of, wow, they spent 1 month in space, and that was really exciting. We've reached the point, we had the 1-year mark with Scott Kelly, and we've reached the point now where we're actually setting up regular cadence of 1-year missions with the astronauts. The fact that we can do that comfortably and they can live and work in this environment as if it's second nature was groundbreaking. I would say that using the platform, from our perspective in the sciences and the research area, using that platform to help understand disease on Earth has been game-changing. It's impactful for our astronauts, and I think that will be another huge legacy and one that I'm focused on in my division moving forward.
Absolutely, thank you. And I'm sure, Stefanie, BioServe has visions of continuing to support life science R&D on the future platform once operational. How can this space station help to inform hardware and integration teams like yours in the remaining years? We have those orbiting platform still available.
I think that it's a big question too. But I think in the remaining years, you know, we can still get a lot more science done that will help, you know, develop the basis for science on the commercial platforms that could generate revenue to help keep the commercial platforms running. So it was mentioned the in-space biomanufacturing, and so we're developing a platform that right now it's a small footprint to fit into a rack on the station, and it's for manufacturing stem cells. But it's being designed so that it can be scaled up, so that on a commercial platform, it can be much bigger. It can be outside of the envelope that we're required to stay within right now on the International Space Station. So I think being able to use the station to, in its last few years, I think there's still a lot of science to do, but I think being able to gear some of that science and hardware development towards those commercial platforms, and so that so that we can help those commercial platforms be successful and continue both applied and fundamental research, I think will be huge.
Yeah, I totally agree. And Arun, what's next for you?
Yeah, a lot of things. The thing that really excites me the most are some of these biomanufacturing applications, you know, figuring out things that we can only do in LEO, biomedical research experiments, discoveries, things that can be enabled by the presence of microgravity. And we're starting to see that. In fact, you know, we had an experiment that we flew together on AX2 where we saw some really cool preliminary results about how three-dimensional stem cell aggregates could actually, the microgravity environment can facilitate the formation of those aggregates. And so these are initial experiments, initial observations, but we think that this potentially has a lot of ground-side potential as well for improving stem cell manufacturing here on Earth. And so that's what's next for me, is to be able to flesh out some of those observations into truly reproducible experiments, ultimately that could lead to commercial benefit for folks here on the ground.
So we have a few minutes left. I have a couple of questions here. I have one here. In regards to fundings, why does space farm R&D lag other space areas? Like why, yeah, I guess the meaning, like, why is covering of the space areas, there's like less funding, is the most voted question.
So, sorry if I can.
In regards to funding, why does space farm R&D lag other space areas? Yeah.
Is it a question for me or for anybody? Or
Who wanna take it?
okay. Why does this area lack funding? I actually don't think it lacks funding.
Right.
I feel like
we
there's
were talking like
quite a bit of funding,
we have
both on the
fundings.
government side as well as the private side. I mean, this conference, I think, is a reflection of that. There's definitely an interest in this area of research. There's fundamental discoveries that are being made, but those fundamental discoveries lead to commercializable products, right? And if that can be identified, then funding will inevitably come.
Cool.
Can I add to that?
Yeah, sure.
You know, when we're thinking about, you know, bringing corporate in, you know, big companies, pharmaceutical companies, you know, a lot of times they're looking at wanting to show that demonstrated proof. And I think we're getting there to that. And so I would say, I think we'll see a lot more of that investment in microgravity research by them in the future.
No, absolutely, I agree with you guys. And there is another question, this is an interesting question for you, Dr. Carnell. What do you think are the current critical gaps in technologies on the ISS in the biological science area?
That's a good one. We do have, as far as the technologies, or I would say maybe capabilities that we're actually able to accomplish that end-to-end science, we are missing some of the key hardware that you need from your bench, you know, to actually complete a full experiment on the mission. So even if, you know, we have a CIRRUS program that we've been kicking off, and you might have heard about that, commercially enabled rapid space science, where we are looking to send that scientist astronaut in space, do the end-to-end experiment, and come home with a complete analysis without having to launch back and forth. That will bring a lot of benefit to the pharmaceutical industry and the commercial LEO economy. But for those pieces of hardware, there are so many things missing. You know, I think we could use a microplate reader as a basic one. You know, and so looking through the lab of what facilities we have up there, what you need to do to conduct your experiment. And I think bringing forward that hardware in your proposal is of value, because you never know, we may be able to actually accommodate that or find a way to get something flown.
And there is a burning question here, and I think we have time for this, and that's it. Why ISS being deorbited in 2030s? What is next to support an international initiative for research and collaboration in low Earth orbit?
So I think that one might be for me too.
I think it is.
So I do have to say that for those of you who haven't been following the space station, you know, extending the life to 2030 is already, we've continued to extend the life. I believe 24 was our first deadline, then it was 28, now it's 30, because it's still maintaining its existence. At some point, as in all hardware and capabilities, you know, things start to break and it's time for new. And also, as somebody said earlier in the first session, at some point it's not NASA's role anymore. We've been in LEO for an extended period of time. The station's been operational for 20 plus years. It's time for that commercial LEO economy to evolve and take over. And then NASA will become one of many users in that vicinity. And as we look to our exploration goals, we'll rely on the commercial destination companies to actually fill that LEO economy role for us. So
yeah, absolutely.
Hopefully that answered the question.
Yeah, no, absolutely. There were more questions, but unfortunately we don't have time to answer, but please come to us, ask questions. You know, we are here and ready to, you know, talk to you, interact, and whatever question or curiosity you have. So I'd like to thank you, each one of you, the panelists, for being here, and the audience for listening.